Hello, this lesson is lesson 4 of topic B6 Inheritance, and it is for my triple science students only. So if you are studying combined science, you can miss this lesson out and go straight on to lesson 5. Okay, very quickly then, just to start us off, I would like you to describe the process of meiosis. You can use diagrams to help you. Give you a couple of clues. I'd like to see the word gametes in there. It would be phenomenal if you could add the words haploid and diploid. And remember, the cells that are made are called daughter cells. As much as you can to describe the process of meiosis, and I'm just going to give you 1 minute. Okay, time's up. So you might have talked about one parent cell doubling its chromosome number for a short period of time, then splitting into 2 cells that each have the diploid number of chromosomes, but then splitting again into 4 daughter cells that have a haploid number of chromosomes. Meiosis produces gametes. The other key difference between meiosis and mitosis is in meiosis there are 2 episodes of cell division. So I've just gone over that really quickly just to remind you about meiosis. I'll give you 10 seconds just to look at the diagram just to help you out if you've forgotten anything from this lesson. Okay, our learning outcomes for this lesson are to describe the structure of DNA, and this is in a little bit more detail than last lesson, which was for combined students only, and to explain the impact of mutations on the synthesis of proteins. So again, you've seen this slide before. I'm just going to give you 1 minute just to write down on your piece of paper the 5 words that go under the white boxes, just to get your brains going and remind yourself of some of these key language words that are important for genetics. Okay, awesome. So, box number 1, you should have had a cell. And last time when we did this, I told you could be forgiven if you've written down cytoplasm, because the end of the black arrow is actually pointing just inside the membrane to the jelly-like substance, which is the cytoplasm. But it was actually referring to the whole cell. Box number 2 is pointing to the nucleus. And you can see inside that nucleus, you've got these blue crosses, which are then enlarged for box number 3, and they are the chromosomes. Box number 4 then is showing a small part of a chromosome, which is made up of DNA. And this term helix talks about the double helix, the 2 strands with the bases across the strands. And finally, a short section of that DNA, a short section of a chromosome, is called a gene. So well done if you got those right. The genetic material in the nucleus of a cell is composed of a chemical called DNA. DNA is a polymer made up of two strands which form a double helix. The DNA is contained in structures called chromosomes. And the way I describe this DNA is if you had a ladder and you held it fixed at the top and you twisted the bottom, you get this twisted double helix, which represents the structure of the DNA polymer. The red and green and yellow and purple things you can see in the diagram join those two upright bits of your ladder. Your phosphate backbones are called the bases. And you can see on that diagram you've got four letters A, G, C and T, and these are the letters which represent the bases. I'm going to look at those in a little bit more detail in a minute. A gene then is a small section of DNA on a chromosome. Each gene codes for a particular sequence of amino acids. And that particular sequence of amino acids then makes up a specific protein. So the order of these bases determines the amino acids which are made. 3 of the bases you can see in the diagram make up one amino acid. And then it's the order of those bases and amino acids which result in the proteins that are actually made. Because proteins are just long chain amino acids made of DNA. The order of those amino acids determines the specific protein that is made. Okay, this diagram is taken from an exam paper and it shows how the genetic diagrams can look if you actually get a question in your exams. So diagram 1 shows an animal cell and some of the structures inside that cell. I want you just to have a think and I'm going to give you 1 minute to decide which word from the box you could use to label structures A, B and C. So 1 minute, see what you can work out. Okay, time's up. So quite simple. The cell on the left, the circle in the middle would be the nucleus. That nucleus is then enlarged and B is pointing to these lines inside the nucleus, which would be showing the chromosomes. And then one of those chromosomes is enlarged again and it's pointing to a short section of that chromosome, which would be a gene. So A is nucleus, B chromosome and C gene. So the double helix ladder of a DNA molecule is held together by rungs made from pairs of chemicals called bases. There are 4 types of bases and they are usually identified by their initial letter. So we've got A, C, G and T. Now the colours and the shapes don't matter, but it does help you to remember that there's a specific way that these bases pair together. And if you look on the diagram, you might be able to work it out. So there's 4 bases, A, C, G and T. What do you notice about the diagram about the way those 4 different coloured bases actually pair together? Okay, so they're the names of them. You don't need to know the names. Anidine, cytosine, guanine and thymine. are just where the A, C, G and T come from. For GCSE science, you're fine to just use the first letters. For A level, you will be expected to know the names. But in terms of how they pair, there are certain rules. So these base pairs are what hold the two strands of DNA together. And there's rules for how they pair together. So the A will always pair with T, and C will always pair with G. So you would never get A and C, or T and G, or C and T, or G and A. It just doesn't happen. A always pairs with T, T always pairs with A, and C always pairs with G, and G always pairs with C. There are millions of base pairs in a single DNA molecule, but they always follow these rules. 4 bases, A, T, C and G. A always pairs with T and C always pairs with G. It's a sequence of these bases along a DNA molecule that forms the specific genetic code. Okay, so a way to remember them. Adidas trainers, Galaxy chocolate, A and T, C and G. You might want to come up with your own. Just a simple way of remembering them. Okay, so what I want you to do, I'm going to give you 3 minutes. I want you to complete the complementary strand of DNA. Now just imagine those black letters all in a row are amino acids. So 3 bases make up an amino acid. So you've got 1, 2, 3, 4, 5, 6, 7, 8 amino acids arranged in that particular order, making up a strand of DNA. And on the complementary strand, the opposite strand, which bases would pair with the bases you have been given. The first one's been done with you. So the first base you've been given was a T. So its base pair must be an A. I'm just going to give you 3 minutes to come up with all the bases on the complementary strand of DNA. Looking above, the letter above, what letter pairs with that letter, so therefore what it must be. 3 minutes, time starts now, off you go. Okay, I'm just going to give you another minute. I don't think that time I gave you exactly 3 minutes, did it? So I'm just going to give you one more minute, just to make sure you've finished. Check them all if you think you've done. Okay, you've probably had enough time. So the first one was given, it was an A, because you were given T, and A always pairs with T. Then we had G, so it must be a C. Then we had C, so it must be a G. So the first amino acid, the first 3 base pairs are AT, CG, and GC. The second amino acid then, you were given CCG, and you should have come up with GGC. The third amino acid, CAG, so you should have come up with GTC. The fourth amino acid, AAT, so you should have come up with TTA. The fifth amino acid, ATA, so you should have come up with TAT. The sixth amino acid, ACG, so you should have come up with TGC. The 7th amino acid, GTA, and you should have come up with CAT. And last but not least, the final amino acid, GTC, and you should have come up with CAG. Because A always pairs with T and C always pairs with G. So you've got a complementary strand of DNA. They're your base pairs, which would hold that DNA double helix together. Proteins are synthesized on ribosomes according to a template. Now, the word synthesis or synthesized is used a lot. Photosynthesis is a good example. It just means made. If you synthesize something, you are just making it. So photosynthesis, light photosynthesis make, plants make their own food using light. Proteins are made on ribosomes. Ribosomes are cell organelles that are found in the cytoplasm. They are the location for protein synthesis. And you would have learnt about the function of ribosomes in B1. So proteins are synthesized on ribosomes according to a template. Carrier molecules bring specific amino acids to add to a growing protein chain in the correct order. Now, you don't need to worry about the carrier molecules and what we call them or how they work. You just need to know that they bring these specific amino acids to the ribosomes in the cytoplasm and add them to the growing protein chain. So what you get is you get a protein chain that is unique. So that particular ribosome synthesizes the particular protein that that cell needs. It's not random. So the protein chain is complete. It has a unique shape. The reason why it has to have a unique shape is you need to be producing proteins that are going to do a specific job, such as an enzyme, a hormone, or producing proteins that can form structures such as collagen. It wouldn't be any good if this ribosome was just knocking out all these random proteins and the proteins being produced didn't have the correct order of amino acids to do the job that needed doing. So in order for cells to function properly, specific amino acids are assembled into specific proteins which have a unique shape and can therefore do a very specific job. It's by no means random. Okay, this slide just summarises lots of the points we've already made as a diagram. So at the top, you've got a combination of the 4 possible bases, A, T, C or G. And you can see those are arranged in triplets or threes to make up amino acids. Those amino acids are then arranged as a long chain to form a protein. So bases have a particular sequence. They code for amino acids. The order of those amino acids arranged on the chain determines the protein that is produced. Okay, this slide is just a recap of the previous 2, just to see what you can remember. So I've given you all 4 statements again, but in each statement I have taken some words out and left some gaps, and I'd like you to see if you can work out what goes in those gaps. You may want to write out the whole thing. You may just want to write numbers 1 to 6 on your paper and write down the words that go in the 6 gaps. I'm going to give you 1 minute. If you do want to write out the whole thing, you might just need to pause the video to give yourself a bit of extra time. Off you go. Okay, time's up. So, proteins are synthesized on ribosomes according to a template. Carrier molecules bring specific amino acids to add to the growing protein chain in the correct order. When the protein chain is complete, it folds up to form a unique shape. This unique shape enables the proteins to do their jobs as enzymes, hormones, or forming structures in the body, such as collagen. And just to give you a little bit more information, collagen is just a protein that provides structure to many parts of your body, including the bones, skin, tendons, and ligaments. So it's just a type of protein that's often used in connective tissues, and it's got good structural properties. So number 1, ribosomes, 2, amino acids, 3, unique, 4, unique, 5, job, and 6, collagen. Okay, so what are proteins made from? Is the next thing I want you to think about. Where are they synthesized? And give the function of proteins in the body. So what are proteins made from? Where are they synthesized? And give the function of proteins in the body. And I'm just going to give you 30 seconds just to jot something down for each of these questions. Okay, so proteins are made from DNA. They are synthesized in the ribosomes and functions, well, they have a unique shape which enables them to do their job as enzymes, hormones, or forming structures in the body such as collagen. So this slide just gives you the real bare bones of the previous slide, just as some revision points. So proteins made of DNA synthesized in the ribosomes, they have a unique shape which enables them to do a unique job, either make an enzyme, make a hormone, or form structures such as collagen. Okay, moving on for a second then, and thinking about mutations. Mutations occur continuously, and a mutation is just a mistake in the way a protein is made. Something isn't quite done correctly or a mistake is made. Often mutations can occur and they don't alter the protein itself. They just alter it slightly, a couple of bases or a couple of amino acids, so its actual appearance or the function of it doesn't change. But this isn't always the case. Sometimes mutations can have quite big impact on individuals. Now what happens is a normal gene copies itself, the proteins are synthesized in the ribosomes, and a normal protein is made. But if for some reason some of the bases in that gene are mutated, an abnormal protein can be made or no protein at all can be made. So either the new code, the mutated code, doesn't code for anything at all, or it codes for a protein that isn't normal, that has significant differences to the protein that actually should have been made. Now, the issues surrounding mutations can be huge, can be very small. But what can happen is an abnormal protein can lead to a defect or a disorder, or in a bigger scale, a genetic disorder or a genetic disease. Okay, so I just want you to think about those mutations for a second. And I want you to think about how such a change in DNA caused by a mutation may result in a change in the protein being made. And I'm just going to give you 1 minute to see what you come up with. Okay, time's up. You might have found this a little bit tough now. If you did, don't worry. Okay, so let's have a look at where we're going with this. When a gene codes for a particular protein synthesized in the ribosomes, the gene is said to be expressed. Most of the DNA code does not actually code for a protein. Remember, about 21,000 genes and 3 billion pairs of bases in any one person. So scientists are still trying to work out what this non-coding part of DNA does do. But what they think it does is it switches genes on and off. So variations in these non-coding parts of the DNA affect how the genes are expressed. The non-coding DNA helps us to answer the question about how we can synthesize so many different things with so few genes. Each gene can control the synthesis of many, many different proteins. This depends on how much of a gene is switched on and how much of the gene is switched off, or which other genes are switched on or off at the same time. Scientists think that variations in non-coding areas of our DNA does actually affect which genes are expressed and how they are expressed. This in turn affects the phenotype, the physical appearance of an organism. So when a mutation occurs, and if we think about mutations in horror movies, they're always linked to something bad, a dramatic event or a big change. The truth in genetics is a bit less exciting. So what it is we are here as a result of millions and millions of mutations that have happened over years and years and years, since the earliest days of life on Earth. New forms of genes result from changes in the existing genes, and these changes, sometimes called mistakes, are just called mutations. They're often so tiny that it's just a change to a couple of bases or the sequence of the order of bases, and we don't really notice them. But over generations, they can result in a gradual change. When mutations take place in the coding DNA, most do not alter the protein at all, or the change is so slight that you might not even notice it. A few mutations can code for a change in an amino acid, which can result in a slightly altered protein that folds into a slightly different shape, and as a result, the protein may be changed in a very small way. So mutations don't often make massive changes as you'd think they would from the horror movies. Often the changes are slight. And when they take place in the non-coding parts of the DNA, they don't directly affect the phenotype. So the genes expressed aren't directly affected. So you almost wouldn't notice that a mutation had happened. However, variants in the non-coding DNA can affect which genes are switched on or off. By changing the genes expressed, changes in the non-coding DNA can have an effect on the phenotype. This is a little bit of extra information about how proteins are synthesized for triple science only. So just to summarise then, not all the parts of DNA code for proteins. Non-coding parts of DNA can switch genes on and switch genes off. So variations in these areas of DNA can affect how the genes are expressed. Mutations can occur continuously. Most don't alter the protein or alter it so slightly the function is not affected. A few mutations, though, can alter the protein, giving it a different shape and therefore altering its function. This may be an advantage or may be a disadvantage. Okay, plenary quiz time. I'll just ask you a few questions and I just want you to jot down your answers. I'll give you about 10 seconds each time. So question 1, whereabouts in the cell do you find the chromosomes? Okay, brilliant. I'm sure you've got this one right. It is in the nucleus. Question 2. DNA is a polymer made up of 2 strands that are twisted to form a double what? 10 seconds. Okay, I'm sure you find this one easy. A double helix. The ladder that's just been twisted from the bottom is called a double helix. Third question. The rungs of the ladder, like structures, are called what? So the cross pieces that join the 2 backbones together are called what? Okay, I'm sure you got that one as bases. Question 4, how many different bases are there? And if you could give me an indication of what they are, that would be great. Okay, so we've got 4 different bases. A and T, C and G. And for those of you who are really on the ball, you might have told me that A always pairs with T and C always pairs with G. Okay, question 5 then, going back to last lesson. What is the human genome and why is it useful? And I'm going to give you a little bit longer for this one because there's 2 parts to it. So what is the human genome and why is it useful? Okay, so the human genome is the entire genetic material of a human. It's useful because it can give us information that can help us find causes and research any potential treatments for inherited disorders, as well as tracing any migration patterns from the past. Question 6, define the term gene. Okay, easy one. So a short section of a chromosome, and again they're made of DNA. And finally, the last task for this lesson, I've just given you the notes. So we've got a little bit on DNA structure, a little bit on protein synthesis. If you just want to pause the video now and get those notes down, and then you can have a read through them before you do the next lesson or in a few days to see what you can remember. I'm just going to read through them with you and then you can pause the video. So a DNA molecule consists of 2 sugar phosphate strands connected together by base pairs. There are 4 different bases, G, C, A and T. Base C always pairs with base G and base T always pairs with base A. Protein synthesis. Proteins are synthesized, made on ribosomes. Carrier molecules bring specific amino acids to add to the protein chain in the correct order. When the protein chain is complete, it has a unique shape which enables it to carry out its particular function. Enzymes, hormones, and other body structures are made up of protein. So if you just want to pause the video there and get those notes down, just make 2 nice summaries, one on DNA structure and one on protein synthesis. And then moving on to the last slide, again, I've just given you some summary notes. So what is the human genome? The human genome of an organism is the entire genetic material of that organism. Why is knowledge of the human genome useful? Allows links to be made between genes and different diseases, increases understanding so we can treat inherited disorders, and allows us to trace human migration patterns from the past. And again, you might want to pause the video now and get those notes down as a summary of the human genome and the benefits of studying it. And that is the end of this lesson. Well done for getting through. Some difficult things covered, but if you just want to go back, watch the lesson again, then it might help you just consolidate your understanding.